into how aluminum impairs methylation. Methylation is critical for everything we do, for every cellular function. This is when a methyl group, or CH3, is transferred to DNA, RNA, enzymes, proteins, neurotransmitters, everything. And this is happening in your body billions of times per second in our cells. It is essential for life, and when we aren't methylating effectively or efficiently, we get toxin buildup, fatigue, mental health conditions, hormones out of whack, genes turned on or off that shouldn't be, weak immune systems, high homocysteine that is linked to heart disease. I just wanted to review this so that everyone understands the necessity of methylation. If we look at my graphic, studies have shown a lot of ways that aluminum impairs methylation. The first one we're going to look at is inhibiting DNA methyltransferase. So DNA methyltransferase, or DNMTs, are enzymes that are critical for epigenetic regulation. And epigenetics is how your genes are turned on or off, or the coding that tells our bodies what to do. So aluminum inhibits DNMTs by downregulating their activity, mainly in the brain. So this study shows that aluminum exposure in vitro to neural cells of mice decreased DNMT1 and DNMT3A in neurons and increased DNMT3B, demonstrating that aluminum modulates the expression of DNMTs, impairing neuronal survival. You can see in figure E the hippocampal neurons where there's the control versus exposed to 200 micromoles per liter of aluminum, which decrease the dendrite lengths and intersection. We will next look at impaired histone methylation. Now we're looking at how aluminum impairs histone methylation. So histones are what keep our DNA compact to fit into a nucleus, and they can be modified through methylation as well as acetylation and phosphorylation. Methylation can cause DNA to tighten or loosen. These are our on and off switches to genes. So if you can't actively methylate certain histones, you get decreased expression of genes for things like DNA repair and neuroplasticity, or you may get activation of genes that are typically silenced, like cancer genes. So disruption of histone methylation is linked to cancer and developmental and inflammatory disorders. There's actually dysregulation of H3K27Me3 and several histone methylation enzymes that are observed in autism spectrum disorder. Aluminum interferes with the activity of the enzymes that methylate histones. This study shows that aluminum caused a decline of H3K27Me3 and decreased the enzyme activity and expression of MLL, a methylation enzyme. It also showed that aluminum reduced the activity of histone methylate, demonstrating that chronic aluminum exposure may inhibit PHF8 and prevent it from functioning or blocking demethylation, and decreasing BDNF protein expression, impairing learning, memory, and neuroplasticity. I know this gets confusing when there is a ton of abbreviations like H3K27Me3 or PHF8 or BDNF. So I added some information about these, but it's always good to look them up and do your own research just for better understanding. Or if you have questions, drop them in the comments. Continuing on how aluminum impairs methylation, specifically by inhibiting methionine synthase. For anyone new to my page, I'm Michaela. I'm a pharmacist and mom wanting better for my kids' health and futures. So I'm researching and sharing that education with you from scientific literature. Diving in, methionine synthase is a key enzyme in the methylation cycle. It converts homocysteine to methionine, regenerating methionine to donate methyl groups, which are critical for one-carbon metabolism. It requires B12 as a cofactor and is highly sensitive to oxidation, and methionine synthase activity affects all methylation reactions. Methionine is a precursor of SAM, a primary methyl donor of more than 100 enzyme reactions. Decreases in methionine synthase increase homocysteine, and high homocysteine increases risk of neurological and cardiovascular disease, and really all functions because methylation is affected. This study showed that pre-treatment with aluminum as well as other metals lowered glutathione levels and inhibited methionine activity. You can see in figure A the methionine synthase activity decreased with aluminum concentration. It also decreased uptake of cysteine, which is the rate-limiting step for glutathione synthesis. You can see in figure D with the aluminum having lower cysteine uptake. So we go back to the graphic and we see that aluminum impairs methylation by inhibiting methionine synthase, reducing methyl groups and increasing homocysteine, inhibiting methylation reactions and leading to global hypomethylation. Follow me to learn more on aluminum's impact on methylation. How does aluminum impair methylation through microRNAs? Anyone new to my page, I'm Michaela. I'm a pharmacist and mom wanting better for my kids' health and future, so I'm researching and sharing the education with you from scientific literature. First, microRNAs are short RNA molecules that are critical in regulating gene expression. They bind to messenger RNA, and that can reduce how much protein is produced, affecting various functions. This study demonstrated that aluminum inhibits the IRS1 PIK3-AKT pathway, a signaling pathway that's critical for metabolism, glucose uptake, protein synthesis, gene expression, various other functions, and survival. And it increased A beta and tau in the brain of rats. There was 167 microRNAs with significantly changed expression in the aluminum-exposed rats compared to the control. So you can see in these figures, the red dots are transcripts that were upregulated, and the blue dots are those that were downregulated, with the majority being downregulated. There's also a list of those differentially expressed genes, or DEGs, that are in table 1 here. So for context, changes in the regulation of these pathways can lead to neurodegeneration, increase in cell death, insulin resistance, DNA damage, and many other negative functions. The protein levels of tau increased with increased aluminum. A beta 142 levels increased significantly in the aluminum groups, affecting learning and memory. Overall, it was demonstrated that aluminum inhibits this pathway, resulting in abnormal phosphorylation, which is what we see in Alzheimer's disease when you have the tangles. But you can see how this can apply to any neurodegenerative or neurodevelopmental disease. Follow me to learn more on how aluminum impairs methylation.